Somewhere in the next 20 years, 1 in 3 Americans currently over the age of 65 will develop a form of measurable, clinically significant cognitive decline. Not all of them will receive a diagnosis of Alzheimer's disease. Not all of them will progress to severe dementia requiring full-time care. But the trajectory of cognitive aging that the majority of older Americans are currently on, without any awareness that an alternative trajectory exists, leads toward a meaningfully diminished capacity for memory, for decision-making, for the rich cognitive engagement with the world that defines independent living and personal identity. And here is what I need you to understand at the very outset of this conversation, because it fundamentally reframes everything we are about to discuss. The brain you have at 65, at 70, at 75, is not predetermined by your genetics in the way that most people believe. The landmark research in cognitive neuroscience over the past 2 decades has produced one of the most genuinely hopeful discoveries in all of medicine. The discovery that the human brain retains the capacity for structural change, for the formation of new neural connections, and in certain circumscribed regions, for the generation of entirely new neurons throughout the entire human lifespan. This capacity is called neuroplasticity, and it means that the trajectory of your cognitive aging is not fixed. It is, to a meaningful and scientifically documented degree, a function of specific, identifiable behaviors. Behaviors that either actively protect and strengthen the neural architecture of your brain, or behaviors that passively allow that architecture to deteriorate at an accelerated and largely preventable rate. Among the dozens of lifestyle factors that influence cognitive trajectory after 60, there is one specific morning practice, grounded in a robust and rapidly expanding body of neuroscientific research, that exerts a disproportionately powerful protective effect on brain structure, brain function, and long-term dementia risk. It is not a supplement. It is not a puzzle app. It is not an expensive cognitive training program marketed with promises that exceed what the science actually supports. It is a specific, deliberate, physiologically grounded practice that takes approximately 20 to 30 minutes, that costs nothing, that requires no special equipment, and that the overwhelming majority of Americans over 60 are not currently doing, despite the fact that the neuroscience supporting its protective effects has been accumulating in peer-reviewed literature for more than 15 years. Today, I am going to walk you through exactly what this practice is, the precise neurobiological mechanisms through which it protects your brain, and how to implement it correctly starting tomorrow morning. Hello everyone, I am Dr. Kaur, and welcome back to Dr. Kaur Health. What we are covering today represents one of the most consequential and most underutilized opportunities in preventive brain health. A single, well-defined morning practice supported by an extensive body of neuroscientific research, yet almost never discussed with the clarity and the mechanistic depth it deserves in routine clinical practice. This is going to be a long, thorough conversation, because brain health deserves nothing less than the complete scientific picture. We are going to cover the neuroscience of aerobic exercise on brain structure, the specific physiological mechanisms involved, the precise protocol the evidence supports, and how this single morning habit interacts with sleep, nutrition, and cognitive engagement to produce compounding protective effects over years and decades. If you are new here, subscribe and turn on notifications. Settle in. This is a conversation worth your complete attention. Before we can understand how to protect the brain, we need a clear and accurate picture of what is actually happening within the aging brain, because the popular understanding of brain aging is, in several important respects, incomplete or outdated relative to current neuroscience. The human brain reaches its maximum volume in the mid-20s. From that point forward, total brain volume declines gradually, at a rate of approximately 0.2% per year through the 50s, accelerating to approximately 0.5% per year after 60, and further accelerating in the 70s and beyond. This process is called age-related brain atrophy, and it affects different brain regions at substantially different rates. The hippocampus, a seahorse-shaped structure located in the medial temporal lobe, and the primary brain region responsible for the formation of new declarative memories and for spatial navigation, is one of the regions most vulnerable to age-related atrophy. The hippocampus shrinks at a rate of approximately 1 to 2% per year in older adults without dementia, and at a substantially accelerated rate in individuals who go on to develop Alzheimer's disease. The prefrontal cortex, the brain region responsible for executive function, including working memory, planning, decision-making, and impulse control, also demonstrates significant age-related volume reduction, contributing to the commonly observed decline in multitasking capacity, processing speed, and complex problem-solving that many older adults notice subjectively. At the cellular and molecular level, several interconnected processes drive this structural decline. The first is reduced cerebral blood flow. The brain, despite representing only approximately 2% of total body weight, consumes approximately 20% of the body's total resting oxygen consumption and receives approximately 15 to 20% of cardiac output. This enormous metabolic demand requires a continuously high rate of blood flow delivery. After the age of 55, cerebral blood flow declines progressively, driven by the same arterial stiffening and atherosclerotic processes affecting the coronary and peripheral arteries that we have discussed in previous videos, now manifesting in the cerebral vasculature. This reduction in cerebral blood flow has direct consequences for neuronal health, because neurons are extraordinarily metabolically demanding cells with minimal capacity to store energy reserves. They depend on a continuous moment-to-moment supply of oxygen and glucose delivered by cerebral blood flow. Even modest reductions in regional cerebral blood flow can impair neuronal function, reduce synaptic activity, and over sustained periods, contribute to neuronal atrophy and death. The second process is reduced neurotrophic support. Neurotrophic factors are a family of proteins that support the survival, growth, and differentiation of neurons throughout the lifespan. The most extensively studied of these is brain-derived neurotrophic factor, commonly abbreviated as BDNF, a protein that plays a central role in synaptic plasticity, in the survival of existing neurons, and critically in neurogenesis, the formation of entirely new neurons from neural stem cells. BDNF levels decline measurably with age. Lower circulating in brain tissue. BDNF levels are consistently associated with smaller hippocampal volume, reduced cognitive performance, and increased risk of depression and dementia in the aging population. The third process is increased neuroinflammation. The brain contains its own resident immune cells, called microglia, which under normal conditions perform essential housekeeping functions, clearing cellular debris, pruning unnecessary synaptic connections, and responding to injury. With age, microglia shift toward a chronically activated pro-inflammatory state, a phenomenon researchers call inflammaging. This chronic low-grade neuroinflammation contributes to synaptic loss, impairs neurogenesis, and is increasingly recognized as a central driver of age-related cognitive decline and a contributing factor in the pathophysiology of Alzheimer's disease. The fourth process is the accumulation of oxidative stress. The brain's extraordinarily high metabolic rate generates substantial quantities of reactive oxygen species as a byproduct of cellular respiration. While the brain possesses antioxidant defense systems to neutralize these reactive molecules, the efficiency of these defense systems declines with age, allowing oxidative damage to accumulate in neuronal lipids, proteins, and DNA over decades, contributing to cellular dysfunction and, eventually, neuronal death. Understanding these four interconnected mechanisms, reduced cerebral blood flow, reduced neurotrophic support, increased neuroinflammation, and accumulated oxidative stress, is essential because the morning practice we are about to discuss exerts measurable, evidence-documented protective effects on every single one of these four mechanisms simultaneously. The single morning practice with the most robust, most extensively replicated, and most mechanistically well-understood evidence base for protecting brain structure and function after 60 is structured aerobic exercise, performed consistently at a specific intensity, ideally in the morning hours. I want to be precise about what I mean by this, because the term exercise is used loosely in popular health discourse in ways that obscure the specific evidence base we are discussing. The research supporting brain protection through aerobic exercise specifically involves sustained, rhythmic, large muscle group activity, walking at a brisk pace, swimming, cycling, water aerobics, or similar activities, performed at a moderate intensity, meaning an intensity at which conversation remains possible, but somewhat effortful, sustained for a minimum duration of 20 to 30 minutes, performed with a frequency of at least 3 to 5 times per week. This is distinct from resistance training, which we discussed extensively in our video on visceral fat, and which carries its own important and partially overlapping cognitive benefits, and distinct from brief, high-intensity activity, which has a different and less extensively documented evidence base in the cognitive aging literature specifically. The landmark study that established the modern scientific foundation for this relationship was conducted by Dr. Kirk Erickson and colleagues, published in the Proceedings of the National Academy of Sciences in 2011. This randomized controlled trial enrolled adults aged 55 to 80 and randomized them to either a moderate-intensity aerobic walking program or a stretching control group, with brain imaging performed before and after a 1-year intervention period. The results were genuinely remarkable, and they fundamentally changed the scientific understanding of what is possible in the aging brain. The aerobic exercise group demonstrated a 2% increase in hippocampal volume over the 1-year study period. Let that finding register fully, not a slowing of the expected decline. An actual increase in the size of a brain structure that, as we discussed in the previous section, typically shrinks by 1 to 2% annually in this age group. This represented an effective reversal of 1 to 2 years of expected age-related hippocampal atrophy, achieved through nothing more than a structured walking program. The stretching control group, by contrast, demonstrated the expected pattern of continued hippocampal volume decline over the same 1-year period, approximately 1.4% reduction, consistent with typical age-related atrophy rates. This study has since been replicated and extended by numerous subsequent investigations using diverse methodologies, populations, and exercise protocols, with remarkably consistent findings across the literature. A 2018 meta-analysis published in Neuroimage, pooling data from multiple randomized controlled trials, confirmed that aerobic exercise interventions in older adults produced significant increases in hippocampal volume, with effects most pronounced in the anterior hippocampus, the subregion most specifically associated with episodic memory formation. Beyond hippocampal volume specifically, aerobic exercise has been shown across multiple studies to increase gray matter volume in the prefrontal cortex, improve white matter integrity, the structural connections between different brain regions, and enhance functional connectivity within and between brain networks involved in memory, attention, and executive function. The cognitive outcomes associated with these structural brain changes are equally compelling. Multiple randomized controlled trials and longitudinal cohort studies have demonstrated that regular aerobic exercise in adults over 60 is associated with improved performance on tests of episodic memory, executive function, and processing speed, and critically, with reduced risk of progression to mild cognitive impairment and dementia over extended follow-up periods. A landmark study published in JAMA Neurology following a large cohort of older adults over multiple years found that individuals in the highest tertile of physical activity had a 38% lower risk of developing dementia compared to those in the lowest tertile, after controlling for cardiovascular risk factors, education, and baseline cognitive status. Now that we have established the robust evidence base for aerobic exercise as a brain protective intervention, let us examine in detail the specific physiological mechanisms through which this protection is achieved, because understanding these mechanisms reveals exactly why this particular morning practice is so disproportionately powerful. relative to many other commonly recommended brain health interventions. Mechanism 1. BDNF upregulation. Aerobic exercise is, among all known interventions, one of the most powerful physiological stimuli for increasing brain-derived neurotrophic factor production. During sustained aerobic activity, contracting skeletal muscle releases a class of signaling molecules called myokines into the circulation, including a molecule called irisin, produced through the cleavage of a precursor protein called FNDC5. Irisin crosses the blood-brain barrier and directly stimulates the expression of BDNF in the hippocampus. Simultaneously, the increased cerebral blood flow during exercise, combined with the metabolic and mechanical stress placed on neurons during sustained activity, triggers a local increase in BDNF expression within hippocampal neurons themselves through a signaling cascade involving the activation of CREB, a transcription factor that regulates the expression of genes involved in synaptic plasticity and neuronal survival. The functional consequence of this exercise-induced BDNF elevation is substantial. BDNF promotes long-term potentiation, the cellular mechanism underlying memory formation, in which repeated activation of specific synaptic connections strengthens those connections, forming the physical basis of learning and memory. BDNF supports the survival of existing hippocampal neurons by activating intracellular survival signaling pathways and suppressing apoptotic or programmed cell death pathways. And critically, BDNF is one of the primary molecular drivers of adult hippocampal neurogenesis, the process by which new neurons are generated from neural stem cells residing in a specific region of the hippocampus called the dentate gyrus. Mechanism 2. Adult neurogenesis. For much of the 20th century, the dominant paradigm in neuroscience held that the adult human brain was incapable of generating new neurons, that the neuronal population was fixed at the end of early development and could only decline thereafter. This paradigm has been definitively overturned by research over the past 3 decades, which has conclusively demonstrated that the human hippocampus retains the capacity for neurogenesis throughout adult life, including into advanced age. The dentate gyrus of the hippocampus contains a population of neural stem cells that continuously divide and differentiate into new, functionally integrated granule neurons throughout the human lifespan. Though the rate of this neurogenesis does decline with age in the absence of specific stimulating factors. Aerobic exercise is among the most potent known stimulators of adult hippocampal neurogenesis, demonstrated extensively in animal models and increasingly supported by indirect evidence in human studies, including the hippocampal volume increases documented in the Erickson study and its replications. The functional significance of newly generated hippocampal neurons is an area of active and fascinating research. Current evidence suggests that these new neurons play a particularly important role in a specific cognitive function called pattern separation, the capacity to distinguish between similar but distinct experiences and memories, preventing the confusion of related but separate events. This function becomes increasingly important and increasingly impaired with age, contributing to the common complaint among older adults of difficulty distinguishing between similar memories or experiences. Mechanism 3. Angiogenesis and cerebral blood flow. Beyond its effects on neurons directly, aerobic exercise produces substantial and well-documented improvements in the vascular architecture of the brain, directly addressing the reduced cerebral blood flow mechanism of brain aging discussed in the previous section. Sustained aerobic exercise stimulates angiogenesis, the formation of new capillary blood vessels, within the hippocampus and other brain regions, increasing the density of the cerebral microvasculature and improving the delivery of oxygen and glucose to metabolically active neural tissue. Exercise also improves cerebrovascular function through mechanisms parallel to those we discussed regarding peripheral and coronary vascular health in previous videos, improving endothelial function and nitric oxide bioavailability in the cerebral vasculature, improving cerebral autoregulation. the brain's capacity to maintain stable blood flow despite fluctuations in systemic blood pressure, and reducing the arterial stiffness that impairs blood flow delivery to metabolically demanding brain regions during periods of increased neural activity. Multiple neuroimaging studies using techniques including arterial spin labeling (MRI) have documented measurable increases in resting and task-related cerebral blood flow following aerobic exercise interventions in older adults, with the magnitude of cerebral blood flow improvement correlating with the magnitude of cognitive performance improvement. Mechanism 4. Anti-inflammatory effects. Regular aerobic exercise produces a complex and somewhat paradoxical effect on the inflammatory system. Each individual exercise session produces a transient, acute increase in certain inflammatory markers, a normal and adaptive physiological response to the metabolic stress of exercise. But the chronic repeated pattern of this transient inflammatory response, performed consistently over weeks and months, produces a net anti-inflammatory adaptation throughout the body, including within the central nervous system. This anti-inflammatory adaptation involves a shift in the polarization state of microglia, the resident immune cells of the brain discussed earlier, away from the chronically activated pro-inflammatory state associated with aging and toward a more regulated, surveillance-oriented phenotype. Exercise also increases the production of anti-inflammatory cytokines, including interleukin-10, and reduces circulating levels of pro-inflammatory markers, including interleukin-6, tumor necrosis factor alpha, and C-reactive protein, the same inflammatory mediators we discussed in the context of visceral fat in our previous comprehensive video, now demonstrating their relevance to neuroinflammation and cognitive aging. Mechanism 5. Metabolic and insulin sensitivity effects. Given the established relationship between insulin resistance, type 2 diabetes, and increased dementia risk, a relationship robust enough that some researchers have proposed the term type 3 diabetes to describe the metabolic contribution to Alzheimer's disease pathophysiology. The insulin-sensitizing effects of aerobic exercise represent an additional and clinically significant neuroprotective mechanism. Insulin receptors are densely expressed throughout the brain, including in the hippocampus, where insulin signaling plays an important role in synaptic plasticity and memory formation. Insulin resistance, the reduced cellular responsiveness to insulin signaling that we discussed extensively in our previous video on visceral fat, extends to the brain itself, producing a state of central or brain insulin resistance that impairs hippocampal synaptic function and is associated with increased amyloid and tau pathology, the hallmark pathological features of Alzheimer's disease. The same exercise-induced improvements in peripheral insulin sensitivity discussed in our visceral fat video extend to improvements in central insulin sensitivity, representing a direct mechanistic link between metabolic health and brain health that aerobic exercise addresses simultaneously. Having established the robust evidence base for aerobic exercise as a brain protective intervention, I want to address a specific and frequently overlooked question. Does the timing of exercise within the day matter for its cognitive benefits? And specifically, is there a meaningful advantage to performing this practice in the morning hours? The emerging evidence suggests that morning exercise timing confers several additional benefits beyond the core neuroprotective effects of aerobic exercise itself, benefits that are particularly relevant to the specific physiology of aging adults. The first consideration relates to circadian rhythm regulation. The human circadian system, the internal biological clock that regulates the timing of sleep, hormone secretion, body temperature, and numerous other physiological processes, relies heavily on environmental light exposure, and particularly morning light exposure, to maintain proper synchronization with the external day-night cycle. Morning aerobic exercise performed outdoors provides a powerful combined stimulus of physical activity and bright light exposure that strengthens circadian entrainment, helping to maintain the robust, well-synchronized circadian rhythm that becomes increasingly fragile and prone to disruption with age, as we discussed extensively in our previous video on visceral fat accumulation and its relationship to disrupted sleep architecture. This circadian benefit is clinically significant because circadian rhythm disruption itself has been independently associated with increased dementia risk and accelerated cognitive decline in multiple longitudinal studies, likely operating through the same sleep architecture disruption, growth hormone reduction, and cortisol dysregulation mechanisms we discussed previously, now understood to have direct relevance to brain health specifically. The second consideration relates to cortisol physiology. As discussed in our previous video, cortisol follows a natural diurnal rhythm, with levels peaking shortly after waking, a phenomenon called the cortisol awakening response, and declining progressively throughout the remainder of the day. Morning exercise, performed during this natural cortisol peak, appears to work synergistically with the body's natural physiological rhythm, whereas evening exercise, while still beneficial overall, can in some individuals produce a secondary cortisol elevation that interferes with the normal evening cortisol decline necessary for optimal sleep onset and sleep architecture. Given the central importance of sleep quality to the neuroprotective mechanisms discussed throughout this video, including the predominantly nocturnal release of growth hormone and the recently discovered glymphatic clearance system, which we will discuss in detail shortly, preserving optimal evening cortisol decline through appropriately timed exercise represents a meaningful, if secondary, consideration in the overall protective strategy. The third consideration is one of practical adherence. Multiple behavioral studies examining exercise adherence patterns in older adults have consistently found that morning exercise is associated with significantly higher long-term adherence rates compared to exercise scheduled later in the day. This finding likely reflects several converging factors: reduced susceptibility to the accumulation of daily fatigue, decision fatigue, and competing obligations that tend to displace exercise intentions scheduled later in the day, and the establishment of exercise as a consistent, non-negotiable component of the morning routine before the day's unpredictable demands have the opportunity to interfere. Given that the neuroprotective benefits of aerobic exercise are fundamentally dependent on consistency over months and years, a single exercise session, however well-timed, produces only transient and modest effects. The adherence advantage of morning timing represents a practically significant consideration, even though it is not, strictly speaking, a direct physiological mechanism of neuroprotection. One of the most significant discoveries in neuroscience over the past 15 years, and one that adds an additional and compelling dimension to our understanding of why morning aerobic exercise, performed consistently, exerts such powerful neuroprotective effects, is the discovery of the glymphatic system. For decades, neuroscientists understood that the rest of the body possesses a lymphatic system, a network of vessels that clears metabolic waste products, excess fluid, and cellular debris from tissues throughout the body. But the brain was understood to lack any equivalent waste clearance system, a puzzling gap given the brain's extraordinarily high metabolic activity and the corresponding production of metabolic waste products that would, in any other tissue, require active clearance. This puzzle was resolved in 2012, when a research team led by Dr. Maiken Nedergaard at the University of Rochester discovered the glymphatic system, a previously unrecognized waste clearance pathway unique to the central nervous system, named for its dependence on glial cells, specifically a type of glial cell called astrocytes, and its functional similarity to the peripheral lymphatic system. The glymphatic system operates through a remarkable mechanism. Cerebrospinal fluid, which normally surrounds and cushions the brain, is actively pumped into the brain tissue itself along the perivascular spaces surrounding blood vessels, driven by the pulsatile movement of arterial blood flow. This cerebrospinal fluid flows through the brain parenchyma, mixing with and flushing out interstitial fluid containing metabolic waste products, including, critically, amyloid beta and tau proteins, the two proteins whose pathological accumulation defines the neuropathology of Alzheimer's disease. The most striking and clinically significant discovery regarding the glymphatic system is that its activity is dramatically modulated by sleep-wake state. Glymphatic clearance has been demonstrated to be up to 10 times more active during sleep than during wakefulness, driven by a substantial increase in the volume of the interstitial space between brain cells during sleep, a change that allows cerebrospinal fluid to flow more freely through brain tissue and more effectively flush out accumulated metabolic waste. This discovery provides a compelling mechanistic explanation for the long-observed but previously poorly understood relationship between sleep quality and dementia risk. Inadequate or poor quality sleep impairs glymphatic clearance, allowing amyloid beta and tau proteins to accumulate in brain tissue rather than being efficiently cleared, potentially contributing directly to the pathological cascade underlying Alzheimer's disease over years and decades of inadequate sleep. Here is where this connects directly back to our central topic. Multiple lines of evidence suggest that aerobic exercise, and specifically consistent morning aerobic exercise that supports robust circadian rhythm entrainment and optimal sleep architecture, as discussed in the previous section, enhances glymphatic clearance through several converging mechanisms. First, regular aerobic exercise improves the proportion of slow-wave sleep, the deepest stage of non-REM sleep, during which glymphatic clearance is most active. As discussed in our previous video, slow-wave sleep proportion declines significantly with age, and aerobic exercise is among the most well-evidenced behavioral interventions for preserving and enhancing slow-wave sleep in older adults. Second, exercise-induced improvements in cerebrovascular function and arterial pulsatility may directly enhance the pulsatile pumping mechanism that drives cerebrospinal fluid movement through the perivascular spaces, since this pumping mechanism is fundamentally dependent on the pulsatile pressure wave generated by each cardiac contraction reaching the cerebral vasculature. Third, the established anti-inflammatory effects of regular aerobic exercise may help maintain the structural integrity and function of the glial cells, particularly the specialized water channel proteins, called aquaporin 4, expressed on astrocyte end feet, that are essential for normal glymphatic function and which become dysregulated in the context of chronic neuroinflammation. This glymphatic mechanism represents one of the most exciting and clinically significant frontiers in dementia prevention research, and it reinforces a critical message that runs throughout this entire video, that the protective effects of consistent morning aerobic exercise are not limited to its direct effects during the exercise session itself, but extend through its downstream influence on sleep architecture and glymphatic clearance, operating throughout the following night to actively clear the very pathological proteins implicated in neurodegenerative disease. Having established the extensive scientific foundation for this practice, let me now provide the precise, evidence-based protocol for implementing morning aerobic exercise as a brain protective intervention, addressing the specific parameters of intensity, duration, frequency, and progression that the research most strongly supports. Intensity. The evidence most consistently supports moderate intensity aerobic exercise for cognitive benefits, defined using the Borg rating of perceived exertion scale as an exertion level of approximately 12 to 14 out of 20 DARS, corresponding to an intensity at which conversation remains possible but requires some effort. Breathing is noticeably elevated but not labored, and a mild to moderate sweat develops within 10 to 15 minutes. For individuals who wish to use heart rate as an objective intensity marker, moderate intensity corresponds approximately to 60 to 70% of maximum heart rate, with maximum heart rate estimated using the formula of 220 minus age, adjusted as appropriate based on individual fitness level, medication use particularly beta blockers, which blunt heart rate response and physician guidance. It is worth noting explicitly that higher intensity exercise is not necessarily superior for cognitive benefits specifically, and in some studies has shown less consistent or smaller effects on hippocampal volume and cognitive performance compared to sustained moderate intensity activity, possibly due to the differential effects of exercise intensity on the duration and pattern of BDNF release, and the practical reality that moderate intensity exercise can be sustained for longer durations and adhered to more consistently over time by the majority of older adults. Duration and frequency. The protocol used in the foundational Erickson study and replicated across numerous subsequent investigations with consistent results involved 40 minutes of aerobic exercise performed 3 times per week for a total weekly aerobic exercise volume of approximately 120 minutes. This aligns closely with the broader physical activity guidelines published by the American Heart Association and the World Health Organization, which recommend a minimum of 150 minutes of moderate intensity aerobic activity per week for general health benefits, distributed across at least 3 to 5 sessions. For individuals beginning this practice without an established exercise habit, a gradual progression is strongly recommended, both for safety and for long-term adherence. Beginning with 10 to 15 minutes of brisk walking 3 times per week and progressively increasing duration by approximately 5 minutes every 1 to 2 weeks as tolerated, until reaching the target of 30 to 40 minutes per session, represents a physiologically sound and behaviorally sustainable approach to building this practice. Modality selection. While brisk walking represents the most accessible, most extensively studied, and most strongly recommended modality for this practice, given its minimal equipment requirements, minimal injury risk, and high adherence rates in older populations, the underlying neuroprotective mechanisms discussed throughout this video are not specific to walking and extend to any sustained, moderate intensity, rhythmic aerobic activity. Swimming and water aerobics represent excellent alternatives, particularly valuable for individuals with joint conditions, arthritis, or balance concerns that may limit walking tolerance, as the buoyancy of water substantially reduces joint loading while still providing the cardiovascular and metabolic stimulus necessary for the neuroprotective benefits discussed. Stationary or outdoor cycling provides similar benefits with reduced impact loading compared to walking, representing a valuable option for individuals with lower extremity joint conditions who can tolerate the specific biomechanical demands of cycling. Dancing, particularly structured dance classes that combine aerobic exercise with the cognitive demands of learning and executing choreographed movement sequences, has shown particularly promising results in several studies, potentially reflecting an additive benefit from combining aerobic exercise with the cognitive engagement and social interaction inherent to group dance activities, a combination we will discuss further in the following section. Practical implementation considerations. For individuals with significant pre-existing cardiovascular disease, uncontrolled hypertension, or other relevant medical conditions, consultation with a physician prior to initiating or substantially increasing aerobic exercise intensity is an important safety consideration. And in some cases, formal exercise stress testing may be appropriate prior to beginning a structured exercise program, consistent with standard clinical guidelines for exercise prescription in older adults with cardiovascular risk factors. Outdoor exercise, when feasible and weather appropriate, is preferable to indoor exercise specifically for the morning timing protocol discussed in the previous section, given the additional benefit of natural morning light exposure for circadian rhythm entrainment. However, indoor exercise, whether on a treadmill, stationary bicycle, or in a pool, confers the same core neuroprotective benefits discussed throughout this video and represents an entirely appropriate alternative when outdoor exercise is not feasible due to weather, safety, or personal preference considerations. Consistency across the majority of days in a typical week is more important to the cumulative neuroprotective benefit than achieving a specific exercise intensity or duration on any individual day. The research supporting brain protection through aerobic exercise reflects the cumulative effect of sustained practice over months and years, not the effect of any single exercise session, and individuals should be encouraged to view occasional missed sessions as inconsequential to the overall trajectory of benefit, while maintaining the consistent practice as the central, non-negotiable behavioral commitment. While aerobic exercise performed in isolation produces the substantial neuroprotective benefits documented throughout this video, An important and growing body of research suggests that combining aerobic exercise with simultaneous cognitive challenge and social engagement may produce additive or even synergistic benefits beyond those achieved through aerobic exercise alone. This concept is sometimes referred to in the research literature as combined or multimodal intervention, and it is grounded in the understanding that cognitive engagement and social interaction independently activate complementary neuroprotective mechanisms that, when combined with the exercise-induced mechanisms discussed throughout this video, may produce a more comprehensive protective effect on brain structure and function. Cognitive engagement, the active, effortful use of cognitive faculties including learning new information, problem-solving, and navigating novel environments, independently stimulates synaptic plasticity and has been associated in multiple longitudinal studies with reduced dementia risk, a relationship that has given rise to the concept of cognitive reserve, the theory that individuals who maintain higher levels of cognitive engagement throughout life develop a greater capacity to tolerate age-related and pathological brain changes before clinical symptoms of cognitive impairment become apparent. The specific mechanism through which cognitive challenge confers neuroprotective benefit involves the same fundamental principle of activity-dependent synaptic plasticity that underlies the exercise-induced mechanisms discussed throughout this video. Novel, cognitively demanding activities require the formation and strengthening of new synaptic connections and engage broader and more distributed neural networks compared to familiar, automatic activities, providing a continuous stimulus for the maintenance and strengthening of neural architecture throughout the brain. Social engagement represents an additional and independently important protective factor. Multiple large longitudinal cohort studies have consistently demonstrated that social isolation and loneliness are independently associated with significantly increased risk of cognitive decline and dementia, with effect sizes comparable to other major established risk factors, including physical inactivity and hypertension. The mechanisms underlying this relationship likely include the cognitive demands inherent in social interaction itself, which requires complex processing of verbal and nonverbal communication, theory of mind, and emotional regulation, as well as the established relationship between social isolation, chronic stress, elevated cortisol, and the neuroinflammatory and hippocampal suppressive effects of chronic stress discussed in our previous videos. The practical implication of this research is that structuring the morning aerobic exercise practice in ways that incorporate cognitive and social elements may enhance its neuroprotective benefit beyond the core physiological mechanisms of aerobic exercise alone. This might involve walking with a friend, neighbor, or organized walking group rather than walking in isolation, combining the established social engagement benefits with the core aerobic exercise stimulus. It might involve walking in a varied, navigationally engaging environment, a park with multiple paths, a neighborhood with varied routes, rather than a fixed, repetitive route on a treadmill, engaging the hippocampal spatial navigation circuits that are particularly responsive to the neuroprotective effects of exercise discussed earlier in this video. It might involve combining the walking practice with an audio component that provides cognitive engagement, a language learning audio program, an educational podcast requiring active attention and information processing, rather than passive entertainment alone. It is important to emphasize that these combined intervention strategies, while supported by an emerging and promising body of research, should be understood as potential enhancements to the core aerobic exercise practice, rather than substitutes for it. The foundational evidence for hippocampal volume preservation and increase, discussed extensively in Section 2, is specifically grounded in studies of aerobic exercise itself, and the cognitive and social engagement elements should be understood as complementary additions that may amplify benefit rather than independently sufficient interventions. The neuroprotective benefits of morning aerobic exercise do not operate in isolation from the broader nutritional and metabolic context of an individual's life, and a brief but important examination of the specific nutritional factors that synergize most strongly with the exercise-based mechanisms discussed throughout this video is warranted. The Mediterranean dietary pattern, characterized by high intake of vegetables, fruits, legumes, whole grains, olive oil as the primary fat source, moderate fish and poultry intake, and limited red meat and processed food consumption, has accumulated one of the most robust evidence bases of any dietary pattern for cognitive protection in the aging population, demonstrated across numerous large prospective cohort studies and, increasingly, randomized controlled trials. The mechanistic overlap between the neuroprotective effects of the Mediterranean dietary pattern and the exercise-based mechanisms discussed throughout this video is substantial and worth highlighting specifically. The Mediterranean dietary pattern is rich in polyphenols, plant-derived compounds with potent antioxidant and anti-inflammatory properties that directly address the oxidative stress and neuroinflammatory mechanisms of brain aging discussed in Section 1. The omega-3 fatty acids abundant in the fish components of the Mediterranean pattern, particularly docosahexaenoic acid, or DHA, are a primary structural component of neuronal cell membranes and support synaptic plasticity through mechanisms that complement and potentially synergize with the BDNF-mediated mechanisms of exercise-induced neuroprotection. The specific dietary pattern most extensively studied in combination with cognitive outcomes is the MIND diet, a hybrid of the Mediterranean diet and the DASH diet for blood pressure management, specifically designed and validated by researchers to optimize brain health outcomes. The MIND diet emphasizes green leafy vegetables, other vegetables, nuts, berries specifically, given their particularly high concentration of neuroprotective polyphenols called anthocyanins, beans, whole grains, fish, poultry, and olive oil, while limiting red meat, butter, cheese, pastries and sweets, and fried or fast food. A landmark study published in Alzheimer's and Dementia, following a large cohort of older adults over an average of 4.5 years, found that high adherence to the MIND diet was associated with a 53% reduction in Alzheimer's disease risk compared to low adherence, with even moderate adherence associated with a substantial 35% risk reduction. The specific point of synergy with the exercise-based practice discussed throughout this video relates to the timing and composition of nutritional intake surrounding the exercise session itself. Adequate hydration prior to morning exercise is important given the established relationship between cerebral blood flow and hydration status, with even mild dehydration shown to measurably impair cognitive performance and reduce exercise tolerance in older adults. Pre-exercise nutrition, while a matter of individual preference and tolerance, should generally favor easily digestible carbohydrate and modest protein content if consumed before morning exercise, to provide adequate glucose substrate for the substantial metabolic demands of both the exercising musculature and the brain itself during aerobic activity, without producing gastrointestinal discomfort during exercise. Post-exercise nutrition represents a particularly important window given the established relationship between exercise-induced BDNF elevation and subsequent synaptic plasticity, with adequate protein intake in the post-exercise period, supporting the synthesis of new synaptic proteins and structural elements required to consolidate the neuroplastic changes initiated by the exercise session itself, directly paralleling the muscle protein synthesis principles discussed in our previous video on visceral fat and resistance training, now understood to have a parallel relevance to neuroplasticity and synaptic protein synthesis in the brain. Having established the extensive scientific foundation and the precise protocol for this practice, I want to address directly several of the most common barriers that prevent older adults from initiating or maintaining a consistent morning aerobic exercise practice, because understanding and proactively addressing these barriers is essential to translating the scientific evidence discussed throughout this video into sustained behavioral change. The first common barrier is joint pain or musculoskeletal limitation, frequently related to osteoarthritis or other degenerative joint conditions that become increasingly prevalent with age. For individuals facing this barrier, the modality flexibility discussed in Section 6 becomes particularly relevant. Water-based exercise, in particular, substantially reduces joint loading while preserving the core cardiovascular and metabolic stimulus necessary for the neuroprotective mechanisms discussed throughout this video. Additionally, beginning with substantially reduced duration and intensity, and working closely with a physician or physical therapist to identify appropriate modifications for specific joint limitations, allows the majority of individuals with musculoskeletal conditions to participate in some form of regular aerobic exercise. The second common barrier is fear of falling, particularly relevant given the balance changes and fall risk that increase with age, and particularly salient for individuals who have experienced a previous fall or who have specific balance-related conditions. For individuals facing this barrier, beginning with supervised or assisted walking, using appropriate assistive devices when indicated, choosing well-maintained, even walking surfaces, and in some cases beginning with stationary cycling or pool-based exercise, which substantially reduce fall risk compared to walking on uneven outdoor terrain, represent appropriate strategies for initiating the practice safely. The third common barrier is cardiovascular or other medical concern about exercise safety, an entirely reasonable consideration given the established relationship between exercise and acute cardiovascular events in individuals with underlying cardiovascular disease. Even though the overall cardiovascular benefits of regular moderate exercise substantially outweigh this small absolute risk in the vast majority of individuals. The appropriate approach for individuals with this concern is direct consultation with their physician regarding the safety of initiating or progressing an aerobic exercise program, given their specific medical history, with formal exercise stress testing considered when clinically indicated, rather than avoiding exercise altogether based on generalized concern. The fourth common barrier, and perhaps the most pervasive across the older adult population broadly, is simply the absence of established habit and the competing demands of daily life that tend to displace exercise intentions in the absence of a specific, concrete, scheduled commitment. The behavioral science of habit formation consistently demonstrates that specific, scheduled, identity-linked commitments, establishing morning exercise as a fixed, non-negotiable component of the daily routine performed at a consistent time, ideally linked to an existing daily anchor habit such as the morning coffee or breakfast routine, substantially increase the likelihood of sustained adherence compared to vague intentions to exercise more or to find time for exercise when convenient. The fifth and final barrier worth addressing directly is the simple absence of awareness regarding the specific, robust, mechanistically well-understood neuroprotective evidence discussed throughout this video. Many individuals are generally aware that exercise is beneficial for health in a broad, non-specific sense, without understanding the specific, compelling, brain structure-altering evidence discussed in Section 2, or the detailed mechanistic understanding discussed in Sections 3 through 5. My hope, in providing this level of scientific detail throughout this video, is that a genuine, mechanistically grounded understanding of exactly how and why this practice protects brain structure and function will provide the motivational foundation necessary to translate intention into sustained, long-term behavioral commitment. The brain you will have at 70, at 75, at 80 is being shaped to a meaningful and scientifically documented degree by the choices you make starting tomorrow morning. This is not an exaggeration, and it is not a simplification of a more complex reality. It is the direct, evidence-based conclusion of more than 2 decades of rigorous neuroscientific research, demonstrated across hundreds of studies spanning animal models, human neuroimaging, randomized controlled trials, and large longitudinal cohort studies, converging on a single, consistent, genuinely remarkable finding. The aging brain retains the capacity for structural growth. The hippocampus, the very structure most vulnerable to the pathological processes of Alzheimer's disease, the structure responsible for forming the new memories that constitute so much of what we value about cognitive independence and personal identity, can increase in volume, even in the 7th and 8th decades of life, in direct response to a specific, Identifiable, achievable behavioral practice. 20 to 40 minutes of brisk walking, performed consistently, ideally in the morning hours, 3 to 5 times each week. This single practice activates BDNF production, stimulates the generation of entirely new hippocampal neurons, improves cerebral blood flow through angiogenesis and enhanced vascular function, reduces the chronic neuroinflammation that drives cognitive decline, enhances the glymphatic clearance system that flushes the very proteins implicated in Alzheimer's pathology from your brain tissue each night as you sleep, and when combined with cognitive engagement, social interaction, and a nutritionally supportive dietary pattern, provides one of the most comprehensive, most evidence-based, and most accessible interventions available anywhere in the field of preventive brain health. You do not need expensive supplements of unproven efficacy. You do not need elaborate cognitive training programs marketed with claims that exceed their evidentiary support. You need a pair of comfortable walking shoes, a safe place to walk, and the consistent commitment to make this practice a non-negotiable part of your morning starting tomorrow. The trajectory of your cognitive aging is not fixed. It is, to a remarkable and scientifically documented degree, within your power to shape. I am Dr. Kaur. Thank you for investing this time in understanding your own brain health today. Subscribe to Dr. Kaur Health for weekly evidence-based health education for adults over 50 and 60. Share this video with someone you love who deserves to understand the power they hold over their own cognitive future. And I will see you in the next one.